Proposed that connections from the basal amygdala to the ventral striatum allow the CS-US association to control aversively motivated instrumental behavior. Figure 1. Fig. 1 Pavlovian fear and avoidance conditioning circuits. Avoidance responses are not emotional responses per SA. They are simply responses. An animal can learn to avoid harm by running, climbing, press ing, swimming, or even remaining stationary. The animal learns to do what it needs to do to attain safety. But the same responses could be used to obtain food if the animal is hungry, and those responses are a way to gain access to food. In contrast, in Pavlovian fear conditioning, the CS elicits specific emotional responses, fear, or defense responses. Researchers were much more inclined to discuss Pavlovian conditioning results specifically in terms of fear/defense circuits. Comparative observations. Amygdala areas have been implicated in fear conditioning in a variety of mammals, including rats, mice, rabbits, and monkeys. C. Davis, 1992. Davis et al., 1997, Fanslow and Poulos, 2005, Johansson et al., in Review, Ledoux, 1996, 2000, Marin, 2001, 2005, Pape and Pear, 2010, Rodriguez et al., 2004. This suggests strong conservation of the circuitry within mammals, including ing humans. Indeed, a large body of work implicates the human amygdala in fear condition ing and in instrumental responses like avoidance. Becker et al., 1995, Damasio, 1994, Delgado et al., 2009, Dolan and Volumier, 2003, Giannaros et al., 2008, Labar, 2003, Austel et al., 2008, Phelps, 2006, Phelps and Ledoux, 2005, Whalen and Phelps, 2009, Whalen et al., 2004. Thus, damage to the amygdala in humans prevents fear conditioning from occurring, and functional imaging studies show that activity increases in the amygdala during fear condition ing. Additionally, a number of studies have found amygdala activation in response to angry or fearful faces, considered to be unconditioned threat stimuli. Adolphs, 2008. Thus, findings involving both lesion studies and functional imag. ing suggests strong correspondence with the animal literature, at least at a gross anatomical level. Techniques available for studying the human brain do not allow precise localization of specific nuclei, though some progress is being made in this regard. Bach et al., 2011, Davis et al., 2011. An important question concerns the nature of the amygdala in non-mammals and the role of the homologous structure in fear conditioning. According to classic view, areas such as the amygdala, being paleomammalian structures, should not exist in reptiles. However, in the 1970s, Cohen, 1975, claimed to have identified the amygdala in avian species and found that lesions of this region disruptive of Pavlovian fear conditioning in pigeons. More recently, there has been much debate about what constitutes the amygdala, and specifically individual amygdala nuclei, in reptiles and birds. Bruce and Neary, 1995, Carton, 1997, Lanuza et al., 1998, Martinez, Garcia et al., 2002, Moreno and Gonzalez, 2007, using connectivity patterns established in 436 mammals. Areas believed to be homologous to the lateral and central nucleus have been identified in lizards. Lanuza et al., 1998, Martinez Garcia et al., 2002. When threatened, these animals undergo tonic immobility, and damage to the CE homolog interferes with this defensive response. Davies et al., 2002. Much more work is needed to resolve what constitutes the amygdala in non-mammalian vertebrates and to determine whether the functions of the amygdala known to exist in mammals have some relation to the function of the homologous regions in the vertebrate ancestors of mammals. Sensory cortex. Sensory thalamus. CS. LA. Amygdala. Circuits mediating threat-elicited reactions and threat-motivated actions. Ventral striatum. BIT. CGL. VP. CE. Emotional evolution in perspective. Emotional amygdala contributions to other amygdala areas have been implicated in Davis et al. 19. Much more work is needed to resolve much more work is 436 mammals. Areas believed to be homologous to the lateral and central nucleus have been identified in lizards. Lanuza et al., 1998, Martinez Garcia et al., 2002. When threatened, these animals undergo tonic immobility, and damage to the CE homolog interferes with this defensive response. Davies et al., 2002. Much more work is needed to resolve what constitutes the amygdala in non-mammalian vertebrates and to determine whether the functions of the amygdala known to exist in mammals have some relation to the function of the homologous regions in the vertebrate ancestors of mammals. Sensory cortex, sensory thalamus, CS, LA, amygdala, circuits mediating threat-elicited reactions and threat-motivated actions. Ventral striatum, BITCNEDACH, 5HT, CGLHPDN, freeze, react, leap, instrumental actions, CE. Figure 1. Fear is the emotion most thoroughly understood in the brain. Much of the progress made has involved studies of Pavlovian fear conditioning in rats. During conditioning, the conditioned stimulus, CS, usually a tone, and the unconditioned stimulus, US, usually a Fuchs converge in the lateral nucleus of the amygdala, LA, to induce synaptic plasticity of the CS inputs, CS, US convergence not shown. The CS is then able to flow through amygdala circuits to the central nucleus, CE, to control the expression of hardwired, automatic, defensive reactions, freezing behavior, autonomous nervous system, ANS, activity, and hormonal release. CE outputs also activate networks in that control the release of neuromodulators, such as norepinephrine, NE, dopamine, DA, acetylcholine, ACH, and serotonin, 5HT, throughout the brain. These, like hormonal feedback, help add intensity to and prolong the duration of the aroused state. In addition to these various automatic responses controlled by CE, the LA also sends information via the basal nucleus, B, to the ventral striatum, especially the nucleus accumbens. The latter connections are likely to be involved in the invigoration of goal-directed behaviors that allow the organism to act in certain ways on the basis of past instrumental learning or on-the-spot decisions about how to cope with the threat. Other abbreviations, ITC, intercalated nuclei of the amygdala, CG, central gray, LH, lateral hypothalamus, PVN, paraventricular nucleus of the hypothalamus, VP, ventral pallidum. 437. Amygdala contributions to other emotions. While the contribution of the amygdala to fear has been most thoroughly studied, it is clear that the amygdala contributes to other emotional states as well. A relatively large body of research has focused on the role of the amygdala in processing of rewards and the use of rewards to motivate and reinforce behavior. Cardinal et al., 2002, Everett et al., 1999, Holland and Gallagher, 2004, Murray, 2007, Nishijo et al., 2008, Salzman et al., 2007. As with aversive conditioning, the lateral, basal, and central amygdala have been implicated in different aspects of reward learning and motivation, as well as drug addiction. The amygdala has also been implicated in emotional states associated with aggressive, maternal, sexual, and ingestive eating and drinking behaviors. Bahar et al., 2003, Galaverna et al., 1993, Mixek et al., 2007, Befaf, 2005, Siegel and Ettinger, 1981. Less is known about the detailed circuitry involved in these emotional states than is known about fear. Emotional evolution in perspective. There is no shortage of theories that have speculated about the relation of emotion circuits to brain evolution. In the tradition of Darwin, basic emotions theorists have proposed that certain emotions are innate, in part because they are expressed the same in people around the world. Buck, 1981, Ekman, 1977, 1992, Izzard, 1971, 1992, Plutchik, 1980, Tompkins, 1962. These innate emotions are said to be mediated by affect programs in the brain. An affect program, in effect, is psychological description of a dedicated neural circuit. Some neuroscientists have adopted the basic emotions idea and have proposed SPE-specific circuits for different basic emotions. Panksepp, 1980, 1998, 2005, though the basic emotions discussed do not completely correspond with those proposed in the psychological theories. The above discussion of the amygdala and its role in fear and defense might be construed as a mini version of basic emotions theory, a version focused on one basic emotion. However, there is a fundamental difference between the approach I take and the approach of basic emotions theorists. The goal of basic emotions theories is to understand subjective states of conscious experience that humans label with emotion words, fear, love, sadness, joy, etc. Their goal is to understand feelings. This is also true of brain science theories of emotion focused on basic emotions. Panksepp, 1980, 1998, 2005, for example, searches for brain systems in animals that underlie feelings in the animals as a way of understand the brain systems that underlie human feelings. Vocalizations that result from tickling a rat are ways of indexing joyful or pleasurable feelings in the rat brain, and freezing, flight, and fight behaviors are markers of fearful feelings. The approach I take is quite different. Ledoux, 1984, 1996, 2002, 2008. I use emotional behavior as a means of indexing circuits that have evolved to allow organisms to deal with challenges and opportunities in their environments. I make no assumption about what an animal is feeling, since I believe it is not possible to scientifically measure, and thus not possible to research, feelings in animals other than humans. I do not deny that other animals may have feelings. I simply question whether these can be studied using scientific methods. Beyond this methodological barrier, I am also critical of attempts to equate feelings in humans and other animals for other reasons. First, most studies that have explored conscious experience in humans have found that when information, including emotional information, reaches awareness, the dorsolateral prefrontal cortex is active, and if information is experimentally prevented from reaching awareness, this area is not active. For summary, see Ledoux, 2008. The dorsolateral granular prefrontal cortex is a unique primate specialization. Proust, 1995, Wise, 2008, and has features in the human brain that are lacking in other primates. Semendefery et al., 2011. If human. 438. Conscious experience depends on these unique features of brain organization. We should be cautious about attributing the kinds of mental states made possible by these features to animals that lack the feature or the brain region. Second, language is a unique human capacity, and conscious experience, including emotional experience, is influenced by language. The once disputed idea that language and the cognitive processes required to support language functions add complexity to human experience has regained respect. Lakoff, 1987, Lucy, 1997. In the absence of language, experience cannot be partitioned in the same way English speakers can partition fear and anxiety into more than 30 categories. Marx, 1987. The diversity with which nonverbal organisms can conceptualize the world and their experiences in it is thus likely constricted by the absence of language. Conclusion. In sum, basic tendencies to detect and respond to significant events are present in the simplest Syngle cell organisms and persist throughout all invertebrates and vertebrates. Within vertebrates, the overall brain plan is highly conserved, though differences in size and complexity also exist. The forebrain differs the most between mammals and other vertebrates, though the old notion that the evolution of mammals led to radical changes such that new forebrain structures were added has not held up. Thus, the idea that mammalian evolution is characterized by the addition of a limbic system, devoted to emotion, and a neocortex, devoted to cognition, is flawed. Modern efforts to understand the brain mechanisms of emotion have made more progress by focusing on specific emotion systems, like the fear or defense system, rather than on efforts to find a single brain system devoted to emotion. Also, progress has been made in animal studies by focusing on emotion in terms of brain circuits that contribute to behaviors related to survival. Functions. Efforts to use scientific methods identify circuits in animals that might correspond to circuits in the human brain that are responsible for conscious feelings are not likely to succeed since we have no way of scientifically measuring feelings in animals. Conscious feelings are an important topic, but one that is best pursued through studies of humans. References. Adolphs, R. 2008. Fear, Faces, and the Human Amygdala. Current Opinion in Neurobiology. Received. Problems and prospects. Intersection. Gilby. Brain and create. Corresponding. Damasio at. Funding information. Abstract. Feelings are conscious mental events that represent body states as they undergo homeostatic regulation. Feelings depend on the interoceptive nervous system, INS, a collection of peripheral and central pathways, nuclei, and cortical regions which continuously sense chemical and anatomical changes in the organism. How such humoral and neural signals come to generate conscious mental states has been a major scientific question. The answer proposed here invokes 1. Several distinctive and poorly known physiological features of the INS, and 2. A unique interaction between the body, the object of interoception, and the central nervous system, which generates the subject of interoception. The atypical traits of the INS and the direct interactions between neural and non-neural physiological compartments of the organism, neither of which is present in exteroceptive systems, plausibly explain the qualitative and subjective aspects of feelings, thus accounting for their conscious nature. Keywords: non-synaptic transmission, consciousness, feelings, homeostasis, interoception. Introduction. Feelings are key homeostatic devices. They convey crucial information concerning life regulation thanks to a defining characteristic, their concise nature. By portraying functional states of the organism, feelings spontaneously reveal to the mind's owner events occurring in the respective body. Relatedly, feelings such as thirst, hunger, and pain also promote deliberate behaviors aimed at making physiological correct times that contribute to maintaining the integrity of the organism. Thus, feelings add a new layer of homeostatic control over and above older varieties of non-conscious automatic regulation. Of note, the indelible tie between feelings and consciousness is often ignored, possibly due to the unjustifiable assumption that consciousness is a general cognitive property that would have arisen independently. Our position is that consciousness probably began its evolutionary history as an intrinsic component of feelings, and that its physiology and status are best explained in that context. The idea that the mental experiences known as feelings correspond to configurations of visceral and metabolic functions, expressed as VARI. Abbreviations: CNS, central nervous system; INS, interoceptive nervous system; NSS, non-synaptic signaling; PNS, peripheral nervous system. Asians on well-being or sickness was first suggested by authors such as William James and Charles Sherrington, 1-2, and has gained support from modern research in neurobiology and psychology, 3-10. Feelings are mental representations of body states. Some of those body states occur spontaneously as a result of basic homeostatic regulation and generate feelings such as hunger, thirst, or pain, which we designate as homeostatic feelings. But some body states are caused by emotive reactions, such as fear, anger, or joy, which alter homeostatic regulation but are largely prompted by external conditions and result in what we call emotional feelings. 11. Ultimately, whether feelings reflect internally generated or externally prompted homeostatic states, they allow us to experience the conditions of life in the interior of our organism, for example, the vagaries of metabolism and their attendant visceral operations, 12-13, and because feelings have an inherent valence. A positive or negative signal manifest, respectively, by pleasure or discomfort. We are, simultaneously, informed of the success or failure of the homeostatic pursuits within our body and given an incentive to act, or not, accordingly. 14. In this article we describe facts concerning the physiology of feelings that may help us understand how they are generated and, by extension, contribute to elucidating a critical process, the appearance of concise mental states based on physical, organic phenomena. Bioessays. 2021, 43:200261. WileyOnlineLibrary.com/journal/B's Copyright 2021 Wiley Periodicals LLC 1 of 11 https://doi.org/10.202002/B's.202000261152111878 2021 6 downloaded from https://doi.org/10.2002/B's.2020000261 https://onlinelibrary.wiley.com/doi/10.2002/B's.2020000261 by Massachusetts Institute of Technology, Wiley Online Library on December 30th, 2022. See the terms and conditions. https://onlinelibrary.wiley.com/terms-and-conditions. On Wiley Online Library for Rules of Use, OA articles are governed by the applicable Creative Commons license. 2 of 11 Carvalho and Damasio. To a first approximation, the idea that feelings represent physiological states may suggest feelings consist of direct perceptions of the organism's interior, that is, the product of canonical, unidirectional signaling from the periphery of the organism to the central nervous system, CNS. The facts we assemble here, however, run counter to that view. They suggest that feelings rely on a unique and more complex process that differs from that of exteroception. The interoceptive nervous system maps the homeostatic state of the body. A distinct set of neural components is in charge of centrally mapping the wide range of physiological reconfigurations that continuously occur within an organism. This apparatus is known as the interoceptive nervous system, INS, a diverse collection of structures which include nerves and ganglia in the peripheral nervous system, PNS, as well as pathways and nuclei spanning every level of the CNS, For 15, the interoceptive process begins with peripheral sensing of homeostatic changes, whether visceral or humoral, courtesy of a wide range of molecular sensors. These include G-protein coupled receptors, such as the purinergic members of the P2X and P2Y families, and ion channels, such as TRP or piezo 1/2, versatile mechanosensors implicated in a wide range of physiological phenomena, from detecting bladder stretch and coordinating urenatation in the urothelium, 1617, to sensing blood pressure in the aortic arch, 18, to functioning as airway stretch sensors critical for breathing. Regulation, 19, together, this diverse array gathers information spanning mechanical, metabolic, immune, and neuroendocrine parameters from smooth muscle, mucosae, glands, and other viscera throughout the body. 20 to 22, interoceptive signals collected peripherally are transmitted to the CNS for further processing. The vagus nerve and the lamina I, spinothalamicortical, pathway are two prominent conduits of interoceptive information. The INS also comprises ganglia, example, spinal and trigeme NAL ganglia, areas of the central autonomic network and other cortical territories such as the insula, the somatosensory cortices, and the cingulum. 23 to 26, these structures continuously monitor the state of the body and orchestrate responses. Although the INS constitutes a cohesive functional unit playing a fundamental physiological role, there is extensive crosstalk, and indeed overlap, between the INS and other well-described neural systems. As mentioned above, the INS is at once central and peripheral, compris ING numerous structures throughout both the CNS and PNS. Moreover, the INS shares numerous structures with the autonomic nervous system, example, visceral afferents such as the vagus nerve integrate both the interoceptive and autonomic operati, but also extends beyond it. Example, the lamina I pathway and the insular cortex play pivotal roles in interoception, but do not belong to the autonomic network in the strict sense. There are also some similarities between INTE reception and proprioception, and some authors even categorize the latter as an interoceptive sense. 27 to 29, but although both sensory modalities map objects internal to the organism and can thus be contrasted with exteroception, substantial functional and anatomical differences justify separating proprioception from interoception, a distinction first noted over a century ago by Sherrington, 30, and supported by later evidence, 31. Lastly, there is extensive integration of visceral and somatosensory information, a process some authors refer to as somatovisceral afferents. Strikingly, subsets of C fibers originating in specific cutaneous territories mediate effective tactile experiences that have been referred to as sensual touch and project to the insular cortex. 4, 32, 34, blurring the distinction between interoceptive function and the exteroceptive senses. This remarkable instance indicates that the skin, the physical boundary between the interior and the exterior of the organism, is at once internal and external, simultaneously in INTE receptive and exteroceptive territory. 35. In conclusion, the INS is a large collection of neural structures. Some viscerosensory, some somatosensory, some autonomic, some peripheral, some central, that cooperate to produce a real-time map of the homeostatic state of the body. Remarkably, in spite of substantial previous investigations, the structural specifications of the INS components, especially of those located in the PNS, as well as the intricacy of the operations they execute, have not been comprehensively appreciated. Nor have they been discussed in terms of their full neurophysiological significance beyond the obvious fact that they assist visceral and metabolic coordination. Here we suggest that the unusual anatomical structures, physiology, and overall arrangement of the interoceptive system play a critical role in the production of feelings, and, specifically, account for their subjectivity, the fact that they are spontaneously conscious. Interoception is structurally and functionally distinct from exteroception. The interoceptive process shares some features with exteroceptive senses, but is radically different in a variety of ways. Interoception involves signals traveling from the organism's non-neural interior to its neural core, as well as extensive responses and control from the core to the periphery. 29. This latter top-down component is likely to include cortically generated interoceptive predictions reflecting expected body states based on prior experience. 36-37. While the exact role of these visceral inferences has not been entirely elucidated. 38. The existence of ample crosstalk and corrective efferent commands in response to sensed and/or predicted visceral information is widely accepted. 37. Importantly, the relation between the object of perception, the body proper, and the subject of perception, which is generated by the CNS, is unique, since the latter operates entirely within the former, and since both body proper and nervous system can interact directly and continuously. Nothing comparable holds at all for the RELA. Tie-ins between the world external to our organisms and the exteroceptive channels that handle our perceptions thereof. There is no interactive arrangement between the object and the subject of perception in exteroceptive modalities. For example, the visual system cannot directly modify the objects we perceive visually. Moreover, external objects are sensed by specific receptors located in the distal extremity of the 1521188 2021 6, downloaded from https://onlinelibrary.wiley.com/joy/10.0.2/b.2020000261 by Massachusetts Institute of Technology, Wiley Online Library on December 30th, 2022. See the terms and conditions. https://onlinelibrary.wiley.com/terms-and-conditions. On Wiley Online Library for rules of use, OA articles are governed by the applicable Creative Commons license. Carvalho and Damasio, 3 of 11. Table 1. The unique nature of the INS and interoceptive sensing. Interoceptive pathways, INS, exteroceptive/cognitive/motor pathways, predominant fibers, poorly myelinated or unmyelinated, A delta/C, well myelinated, A alpha/A beta, mode of transmission both non-synaptic and synaptic, predominantly synaptic, time scale both slow, S/min/H, and fast, very fast, mu-SMS. Neural processes supported interoception/visoreception, affect, moods, emotions, feelings, movement, fine perception, learning, reasoning, calculation, verbal language, blood-brain barrier, localized. GAPS, continuous main neurotransmitters and neuromodulators, monoamines, dopamine, noradrenaline, serotonin, ACH, neuropeptides, glutamate, GABA. In contrast to canonic exteroceptive, cognitive, or motor pathways, the interoceptive pathways, collectively referred to as the interoceptive nervous system, INS, tend to exhibit poorly myelinated fibers, non-synaptic signaling, gaps in the blood-brain barrier, and slower nerve transmission mediated by specific neurotransmitters. In spite of these distinctions, there is considerable crosstalk and overlap between interoceptive and exteroceptive modalities. For example, some exteroceptive phenomena, example, sensual touch, can be mapped by interoceptive structures. Respective pathway. They cannot interact directly with neural integration centers at multiple levels of the nervous system. In contrast, the INS exhibits multiple direct entry points for interoceptive information along the neuraxis, peripherally as well as centrally. This constitutes a unique physiological design that highlights the distinctiveness of INTE reception, yet it is commonly ignored. The atypical nature of the INS is further documented by the following set of notable features, Table 1. First, the axons of a large number of interoceptive neurons are either entirely unmyelinated or lightly myelinated. For 93940, the lack of myelin insulation greatly increases the exposure of these fibers to the direct influence of molecules present in the extracellular space. Second, the signaling taking place within this particular apparatus is disproportionately non-synaptic, that is, signaling molecules are released for extracellular diffusion unconstrained by synaptic gaps. Third, the interoceptive path ways exhibit conspicuous gaps in the blood-brain barrier, and, as a result, neurons are directly exposed to the chemical molecules circulating in blood capillaries or in the cerebrospinal fluid. This conditine notably applies to multiple levels of the INS, including the spinal and trigeminal ganglia, as well as various integration centers in the CNS. We propose that, rather than simply modulating more modern and well-understood neural processes such as myelinated conduction and synaptic transmission, these unique features of the INS accomplish the following. A. They constitute original evolutionary properties of neural function. B. They remain the standard modes of operation for interoceptive sensing. And C. They make a unique contribution to the productime and experience of feelings. In the pages ahead, we describe these features in detail and consider their implications with the goal of establishing a more comprehensive survey of the foundations of effect than has been available to date. We propose that, as a consequence of the distinctive anatomical and functional features of the INS, the principles and operations underlying the emergence of feelings are fundamentally distinct from those that support typical exteroceptive, motor, and most cognitive processes. Glossary. Homeostasis. The process of maintaining the physiological parameters, example temperature, pH, nutrient levels, visceral operations, of a living organism within the range most conducive to optimal function and survival. The related but distinct term allostasis refers to the specific mechanisms used by an organism as it seeks to regain homeostasis. 41. Emotions. Collections of involuntary actions, smooth muscle contractions, changes in heart rate, breathing, hormonal secretions, facial expressions, posture, etc., triggered by actual or recalled stimuli. Emotions are aimed at supporting homeostasis, for example, countering threats, fear, or signaling success, joy. Feelings. The mental experiences that accompany ongoing homeostatic states of the body. Feelings mentally translate internal organism changes, either spontaneous, for example, hunger or thirst, or caused by emotions, for example, fear, joy, sadness, which are often triggered by extranal events. The genesis of feelings is rooted in specific anatomical and physiological features of interoception, which are fundamentally different from those of exteroception. Interoceptive nervous system, INS. A collection of nerve pathways and CNS nuclei dedicated to detecting and mapping homeostatic signals, such as degrees of visceral muscle. 152118878, 2021, 6, downloaded from https://onlinelibrary.wiley.com/docs/1002/b.202000261 by Massachusetts Institute of Technology, Wiley Online Library on December 30th, 2022. See the terms and conditions. https://onlinelibrary.wiley.com/terms-and-conditions. On Wiley Online Library for Rules of Use, OA articles are governed by the applicable Creative Commons license. 4 of 11 Carvalho and Damasio. Contraction and internal milieu chemical composition. The vagus nerve and the lamina I, spinothalamo-cortical, pathway are prominent examples of interoceptive pathways. The interoceptive system monitors the state of the body and orchestrates automatic responses thereto. Here we propose that it also plays a key role in gene-rat-ing feelings and thus contributing to life regulation at a conscious level. Denuded axons enhance the sensitivity of interoceptive structures. The interoceptive nervous system exhibits a characteristically low level of myelin insulation, 642-47. For example, the lamina I spinothalamo-cortical pathway is composed of small, slow-conducting unmyelinated C and poorly myelinated A delta fibers, whereas the trunk of the vagus nerve, a principal viscerosensory pathway, contains up to 94% unmyelinated fibers. For 39-46-50, the central relays receiving interoceptive signals the area postrema, the nucleus of the tractus solitarius. The parabrachial nucleus in the periaqueductal gray are also poorly myelinated, 51-53. In fact, it has been observed that medial structures of the neuraxis, such as those in the limbic system and brain stem, which are prominently involved in the regulation of homeostasis and in effect, drives, emotions, feelings, are especially rich in unmyelinate fibers, 39-54-55. By contrast, more lateral domains of the mammalian brain, including the main sensoromotor systems, the associa, tine cortices, and the cerebellum, are especially rich in well-myelinated axons, 56-57. Yet another striking example of this dichotomy is found in tactile sensory pathways. Whereas fast-conducting myelinate afferents mediate discriminative touch, unmyelinated C-type fibers thought to belong to the interoceptive system encode a slow and low-discrimination tactile modality referred to as effective touch and project to the insula, 33-34-58. We previously hypothesized that the reason these denuded fibers persisted in evolution, remaining widely represented in complex nervous systems, including that of humans, despite their slow conduction and seeming inefficiency, is their superior sensitivity to circulating molecules. 6. In effect, myelin acts as a physical obstacle and reduces sensitivity. 59-61. Accordingly, unmyelinated axons of the peripheral nervous system are sensitive to extrasynaptic neurotransmitters such as acetylcholine, GABA, serotonin, norepinephrine, and ATP, even when these molecules only reach the axonal trunk, away from the nerve terminals. 42-43-62-64. Moreover, since neurotransmitters can be directly released from denuded nerve trunks, 65-66, unmyelinated fibers are able to both sense and emit circulating molecules, including neurotransmitters. Why would this vulnerability and openness to the surrounding milieu be a desirable attribute in nerve fibers? The explanation may have to do with another ancient and often neglected neural process prevalent in the interoceptive system, non-synaptic signaling. Non-synaptic signaling is prominent in interoceptive structures. The interoceptive nervous system exhibits abundant non-synaptic signaling, NSS. Chemical agents are released into the extracellular space, away from synaptic gaps, and reach their targets by diffusion. NSS is prevalent both in the central and peripheral components of the interoceptive apparatus. Sensory ganglia are prominent examples. NSS has been reported in A. Dorsal root ganglia, DRG, where, although synapses are rare, 67-95% of neurons respond to activation of neighboring cells with an excitatory depolarization, 44, and B. The nodose ganglion of the vagus, 45. Thus NSS may be a general property of neuronal communication in PNS ganglia. In both the DRG and the nodose, NSS is thought to involve non-synaptic chemical mediators, 44-45, but the mechanistic details have remained uncertain. We recently showed that NSS in the DRG involves the purinergic P2RY1 membrane receptor, 68, pointing to extrasynaptic purines. ATP/ADP as critical mediators. Interestingly, P2RY1 appears to act in a cell-type specific manner, promoting NSS in myelinated A-type neurons while inhibiting it in unmyelinated C cells. This dichotomy may play a role in coordinating sensory information transmission and processing. 68. The molecular elucidation of NSS in sensory ganglia opens experimental avenues to test the role of interoceptive processing in the generation of homeostatic body maps and feeling states. Manipulation, inhibition, or stimulation of NSS specifically in ganglia via pharmacological and/or genetic means will allow experimental probing of sensory or affective behavioral changes in model organisms. SPE-specific neuronal subpopulations can be targeted for additional specificity. Moreover, the two major interoceptive pathways, vagal and lamina I spinothalamo-cortical, respectively, can potentially be uncoupled by targeting the nodos or dorsal ganglia individually or in combination, thereby elucidating the role of each branch of the INS. Extrasynaptic neurotransmitter sensitivity has also been reported. Along the axonal trunk of peripheral nerves, for example, in the sural and vagus nerves Wnt with around 42-43, and in the CNS, where the medial structures involved in regulating homeostasis and effect are especially rich in monoaminergic/peptidergic neurons and are presumed to signal abundantly via NSS 54, 55, 69. Neuronal non-synaptic release sites are particularly conspicuous in the area postrema, a chemoreceptor and putative interoceptive integrator, 70, and in vagal projection pathways, 69, among neuropeptides that play a role in effect. Endogenous opioids, such as endorphins, enkephalins, and dynorphins, figure prominently. They bind to and modulate the function of the mu-delta and kappa opioid receptors, 71. Endogenous opioids have been implicated in valence, the qualitative aspect of feelings, which ranges from positive to negative and naturally classifies their homeostatic value as beneficial or detrimental, 72 to 76. All three opioid receptors can be found in extrasynaptic locations, some of them predominantly so, pointing to an important NSS-mediated mode of action of endogenous opioids. 69, 77, 79. Opioids have also been directly implicated in interoceptive. 1521188, 2021, 6. Downloaded from https://onlinelibrary.wiley.com/doy/10.2020/b.2020000261 by Massachusetts Institute of Technology, Wiley Online Library on December 30th, 2022. See the terms and conditions. https://onlinelibrary.wiley.com/terms-and-conditions. On Wiley Online Library for Rules of Use, OA articles are governed by the applicable Creative Commons license. Carvalho and Damasio, 5 of 11. Signaling, in particular in vagal projections to the nucleus of the tractus solitarius, 69, there is remarkable variation in the distance covered by NSS agents acting on interoceptive neurons. In some cases, the signaling molecule is secreted by neighboring cells and travels relatively short distances to reach its paracrine * targets, as happens in some instances of dopaminergic transmission, where the respective receptors have an effective radius of 2 to 8 μm, 80, 2 to 3 orders of magnitude larger than the typical synaptic cleft width, 81. In other cases, signaling molecules cover far longer distances, 82, and can be transported in the cerebrospinal fluid or even in the blood stream. In order to access their target structures, such endocrine signals and metabolites often rely on another peculiar characteristic of the interoceptive apparatus, the presence of gaps in the blood-brain barrier. Blood-brain barrier gaps permit humoral and neural signal convergence in the INS. Another striking feature of the INS is the existence of localized gaps in the blood-brain barrier, BBB, or in the blood-nerve barrier, as it is commonly designated in the peripheral nervous system. The BBB exhibits particularly conspicuous gaps in spinal ganglia, as each ganglion is divided into two regions, one housing most cell bodies and another containing mainly nerve fibers. The former is far more heavily vascularized and shows a dense network of capillaries intimately wrapped around the soma of sensory neurons. In contrast, the nerve fiber-rich area of the ganglion as well as the adjacent nerves are almost entirely devoid of blood vessels, 83. Moreover, the endothelium of the papillar IES supplying the ganglia shows large fenestrations and is permeable to blood-borne agents of high molecular weight. These properties are reminiscent of CNS areas lacking a BBB, such as the area postrema, and have also been reported in other sensory ganglia, example, trigeminal and nodos, as well as in ganglia of the autonomic sympathetic and Enteric nervous systems, 84 to 87, permeable capillaries are also found in the vicinity of nerve roots, in sharp contrast to more distal nerve sections, 85. Blood-borne toxins preferentially affect the sensory pathways, and indeed can accumulate in spinal ganglia, whereas motor pathways are comparatively spared, 83. This selectivity and selective vulnerability is also revealed by intravenous administration of labeled albumin, subsequently found to accumulate in the extravascular, neuronal space of the cell body-rich area of spinal ganglia, but not the nerve fiber-rich area. This firmly indicates that the dense network of capillaries enveloping the sensory and autonomic cell bodies have a specific and topographically circumscribed gap in the BBB, which is presumably a result of the epithelial fenestrations and molecular differences in tight junction composition, 88. Taken collectively, these observations suggest that a lack of BBB is a general feature of the peripheral ganglia carrying interoceptive signals, thus constitutively. Asterisk paracrine signaling involves molecules secreted by a cell and interacting with neighboring cells. Endocrine signaling involves molecules transported in the blood and thus usually cover substantial distances between secretion and receptor binding, exposing these structures to blood-borne proteins and metabolites, in sharp contrast to most regions of the CNS and distal nerve sections. Stripped of a BBB and thus exposed to blood-borne factors, spinal ganglia are likely to operate as chemosensors, responding to blood pH or concentration of metabolites or neuroactive substances, 89, thereby serving as major sites of convergence of neural and humoral signals originating in the body, a role that has typically been associated only with more rostral structures in the CNS. Blood-borne chemical signals crossing blood vessel walls and gaining direct access to neuronal somas may bind surface receptors and trigger changes in membrane potential, thereby having an impact on cellular excitability and action potential propagation, effectively governing the operation of spinal ganglia and, consequently, of interoceptive processing. A unique brain-body interface is the distinctive feature of interoception. The three characteristic features of the INS highlighted above, dearth of myelin sheath, predominance of NSS and gaps in the BBB, are structurally and functionally linked. Indeed, there is topographical overlap of unmyelinated axons and NSS. Unmyelinated, tightly packed fibers are more prone to direct, orthogonal interactions, known as aphasic interactions. An example of NSS, 90, for example, the vagal pathway, one of the most prominent interoceptive channels, is likely to be a key site of NSS. Non-synaptic exchanges, probably neurotransmitter-mediated, have been documented among the cell bodies in the nodose ganglion of the vagus, 45. They may also occur along the vagal trunk, where the predominance of unmyelinated fibers and characteristic dense pack ing offer the ideal anatomical and physiological conditions for aphasic NSS, 691. The subcortical CNS is home to a substantial proportion of unmyelinated structures, and is also the origin of many non-synaptic TIC transmitters, 92, in fact, the same medial regions of the neuraxis. Exhibiting an abundance of unmyelinated fibers also has a high proportion of monoaminergic/peptidergic neurons presumed to signal prominently via NSS 54, 55, 69. As described above, the INS specifically exhibits a high concentration of unmyelinated fibers and abundant instances of NSS, 42, 45, 70. In conclusion, neural structures where NSS is prevalent have consistently large numbers of unmyelinated axons, whereas structures that rely chiefly on canonic synaptic communication, those involved in movement, exteroception, proprioception, and most cognitive processes, are predominantly served by myelinated fibers. The absence of a myelin barrier exposes a denuded neuronal surface, conferring on it the ability to both secrete and respond to signaling agents, that is, the capacity to support NSS. The gaps in the BBB have a similar functional role, exposing neuronal structures to chemical factors present in the bloodstream, as happens in spinal ganglia, or in the cerebrospinal fluid, in the case of circumventricular organs such as the area postrema. The exposed structures gain the ability to sense circulating molecules, detect metabolic changes, and respond accordingly. 152118878, 2021, 6, downloaded from https://onlinelibrary.wiley.com/doy/1002/b.202000261 by Massachusetts Institute of Technology, Wiley Online Library on December 30th, 2022. See the terms and conditions. https://onlinelibrary.wiley.com/terms-and-conditions. On Wiley Online Library for Rules of Use, OA articles are governed by the applicable Creative Commons license. 6 of 11 Carvalho and Damasio. In sum, the dearth of physical barriers, such as BBB and myelin, that is characteristic of the INS enables its unique neuronal sensitive ITY and allows it to interact with physiological compartments, vascular, cerebrospinal, visceral, to which most neurons are not privy. vested with unrestricted access to the physiological interior, interoceptive structures, such as unmyelinated nerves, PNS ganglia, and the circumventricular organs, sit on the frontier between the nervous system and the body proper and effectively constitute an interface where neural and non-neural structures are in direct contact. This brain-body interface is capable of detecting subtle, incipient, and localized chemical changes, thus enabling the mapping of homeostatic states and placing these structures at the very root of interior sensing. We propose that this process is a pivotal contributor to the capacity to experience body states and thus to the emergence of feelings. In support of this view, we note that two of the most distinctive features of interoceptive feel, ING, have striking parallels in the anatomy and physiology of the brain-body interface and the INS in general. First, the characteristic vagueness of some homeostatic feelings, such as well-being, malaise, now see, etc., may be related to the fact that interoceptive pathways are conspicuously exposed and exquisitely sensitive to surrounding modulation not just at their distal extremity, but at every level, from the trunk of peripheral axons to spinal ganglia to CNS nuclei. Thus, the result ING conscious sensation is the summation of myriad factors OPRAT ING at several tiers of the neural apparatus, in stark contrast to typical exteroceptive sensations, for example vision or tactile stimuli, far more direct point-to-point sensory modalities allowing the stimulus source to be clearly and precisely located. Second, the uninterrupted flow of subjectivity may be grounded in the continuity of interoception, 93, reflected in the intimate, continuous contact between the subject and the object of interoception, related to the CNS and the body proper, respectively. Accordingly, spontaneous, slow, continuous discharge has been reported in C fibers and may reflect ongoing tissue metabolic status. 4. The uninterrupted, fluid nature of subjectivity and interoception stands once again in contrast to the typical exteroceptive experience, which is comparatively discrete, with a clearly defined beginning and end, for example, a tactile stimulus. Additional support for the notion that interoceptive sensing is the foundation of affect and subjectivity comes from the field of psychopathology. Numerous psychiatric, neurological, and developmental disturbances involving emotional changes show altered interoceptive processing. Examples include panic disorder, anxiety, depression, autism, and eating disorders. 26, 36, 94. These conditions are associated with structural and functional abnormalities in neural territories play ING interoceptive roles. 38, leading some authors to define depression and anxiety as altered interoceptive states. 95. Neuropharmacological findings are also in general alignment with the idea that the INS is a key neural substrate of feelings. Modulation of visceral function via adrenergic stimulation can produce COR responding feelings and activate interoceptive territories. 96, 97. Moreover, several psychoactive compounds known to influence affective experience have been shown to modulate INS function. For example, PLE3 for methylenedioxymethamphetamine, MDMA, is a monoamine releaser affecting mood, social feelings, and bodily sensations, and new neuroimaging studies show that MDMA administration decreases insular functional connectivity. 98. Changes in insular cortex activity were also reported following treatment with the mood-altering psychedelic drug psilocybin. 99. Similarly, a single dose of ayahuasca, a brew with serotonin receptor agonist properties and documented antidepressive effects, increased blood flow to areas implicated in the regulation of mood and emotions, including the insula. 100,101. Finally, meditative practice impacts affect and alters neural processing of interoceptive information, enhancing some, albeit probably not all, aspects of INTE receptive awareness. 102 to 106. The facts outlined here indicate that the interoceptive brain-body interface plays a central part in sensing homeostatic body states, and we further suggest that it underlies the very nature of feeling. The brain-body interface, evolutionary origins. The brain-body interface consists of phylogenetically ancient structures. Evolutionarily, denuded cells secreting signaling molecules into the surrounding milieu were the norm before myelin, the BBB, and synapses emerged. Coelentrates such as Cnidaria, widely regarded as providing one of the first evolutionary instances of a nervous system, 107,108, commonly exhibit nerve nets composed of denuded neurons. 109. In these organisms, pericardia and neurites are surrounded by other cells, but myelin sheaths are absent. 110. Moreover, cross-species anatomical analysis of the visual system reveals a higher prevalence of unmyelinated fibers in simpler and phylogenetically older organisms, and a gradual increase in myelin insulation throughout evolution, culminating in the very low or absent unmyelinated fibers in the optic nerve of most mammals, including humans. 111. Interestingly, myelin may have evolved independently in different Philadelphia. 109,112,113a. Striking example of convergent evolution. Cnidarian nerve nets exhibit synaptic connections. 107,108. indicat ing that synapses preceded myelin evolutionarily. Interestingly, most neurotransmitters play non-synaptic signaling roles in bacteria, protozoa, and sponges, creatures devoid of nervous systems. 114 to 117. These findings have led to the view that molecules originally serve ing intracrine, autocrine, or paracrine signaling functions in unicellular organisms were later co-opted, with the emergence of multicellularity, to signal between cells of the same organism by diffusion in the extracellular space. 56,118 to 122, with the advent of metazoa and the gradual increase in organism size, evolutionary forces would favor more direct and faster communication. Select ing for elongated cellular projections, ultimately resulting in axons and dendrites. This incremental transition from dispersed chemical release to open synapses and finally, to confined synaptic gaps, would have enhanced the compartmentalization of neurotransmitter transport 123 and increased channel selectivity and speed in point-to-point transmission. According to this view, synapses arose as an evolutionary specialization of paracrine signaling. As for the BBB, a structural separation between the bloodstream and neuronal tissue appears as early as in insects. For example, the fruit 1521188, 2021, 6, downloaded from https://onlinelibrary.wiley.com/doy/1002/bs.202000261 by Massachusetts Institute of Technology, Wiley Online Library on December 30th, 2022. See the terms and conditions. https://onlinelibrary.wiley.com/terms-and-conditions. On Wiley Online Library for rules of use, OA articles are governed by the applicable Creative Commons license. Carvalho and Damasio, 7 of 11. Fly Drosophila melanogaster exhibits a humoral/CNS barrier analogous to the vertebrate BBB that shares some molecular conservation. 124. It has been proposed that the BBB arose as a mechanism to maximize ionic and neurotransmitter stability in the neural interstitium. 125,126. Originally consisting of a glial barrier that later shifted to endothelial tight junctions, freeing glia to specialize in neuronal support and interstitial fluid composition. 127. In support of this scenario, evolutionary vestiges of the glial barrier have been described in the mammalian CNS. 125,128. Each of the three components of the brain-body interface, signaling by extracellular diffusion, denuded axons, and permeable barriers between bloodstream and neural matter, was followed by a functional upgrade, respectively, synapses, myelin, and BBB. Yet the original structures were not entirely replaced and remain well represented in modern nervous systems. The striking longevity of the features of the brain-body interface suggests that they continue to play a fundamental physiological role. We propose that they are essential for interoceptive sensing and the development of subjectivity. The features of the INS may underlie the ability to feel. We have provided evidence for the existence, in the INS, of a distinct set of structural and functional features, namely, abundance of unmyelinated axons, disproportionate reliance on non-synaptic signaling, and conspicuous BBB gaps. These are evolutionarily ancient features that have remained operational and largely unchanged within the interoceptive apparatus. By contrast, within the motor, proprioceptive, exteroceptive, and cognitive systems, the old features have been mostly superseded by A. Myelin insulation, B. Synaptic transmission, and C. A comprehensive BBB. The advent of these traits enabled rapid and precise relaying of neural messages from origin to destination, a critical ability in multicellular organisms with significant organ and system differentiation. We have also called attention to another distinctive feature of interoception. The unique structural and functional features of the INS govern the connection between neural and non-neural compartments of the organism. As a result, the object of interoception, homeostatic body states, and its subject, generated in the nervous system, are in permanent and direct contact, allowing for ongoing mutual modifications. In sum, the INS has both an ensemble of remarkable features and a unique placement within the organism. We propose that these attributes underlie its ability to detect and combine information concerning humoral and visceral body states. Interoceptive axons and INT egration centers, unfettered by myelin sheaths and impermeable epithelial barriers, are open to the direct influence of metabolites and signaling agents that originate in the body proper and can gain access to and commingle in the neural compartment. This arrangement provides fine chemical sensitivity and enables comprehensive and detailed mapping of the changes occurring in the non-neural compartment. In turn, the body can be modified by efferent responses from the CNS, which can also formulate interoceptive predictions based on visceroceptive information, 3638, and modulated by it. The resulting adjustments can be communicated centrally, as part of a bidirectional and interdependent flow. Whether the top-down and efferent aspect of interoception is considered an integral component or a related but separate process, it is clear that extensive reciprocal interactions between the neural compartment and the body proper contribute to the mapping of body states. 2937-129, gating, such as occurs in nociception, 130, is a prominent example of this process. The facts discussed above suggest that ancient structures were selectively retained in the INS because they enable and optimize its fundamental function, interoceptive sensing. Ultimately, the cross-influence between neural and non-neural worlds, facilitated by the atypical INS structures and arrangement, permits the assembling of the graded, qualitative, fluid, and continually body-linked representations we experience as feelings. The findings assembled here have important implications. They respond to well-grounded philosophical critiques of the notion of feelings as straightforward perceptions of body state, 131. They also call attention to the incompleteness of the view that mental processes result exclusively from canonical, synaptic transmission. This notion, expressed in a digital model of mind that informs conventional artificial intelligence and robotics, fails to acknowledge the analog and hybrid processes exploited by interoception, 132. More generally, our findings connect with the recognized need to explain subjectivity. We suggest that the anatomical and physiological features that distinguish the INTE receptive apparatus from the neural systems which support movement and cognition are better candidates to explain the emergence of a RAD. IPLE, unique property of complex, multi-system organisms endowed with a nervous system, the ability to feel body states and naturally engender conscious experiences. Conclusions and outlook. The unique structure and function of the INS set it apart from other major neural systems. Moreover, these traits may help explain the emergence of feelings and some of its characteristic features. Given the central role of affect in health and disease, elucidating the cellular and physiological mechanisms of interoception is of obvious relevance. In particular, experimental exploration of the role of peripheral structures such as the dorsal ganglia should bring a novel perspective to the study of the physiology of feelings. Future therapeutic approaches may be designed to take advantage of the distinctive attributes of the INS. Non-synaptic neuronal transmission is conspicuously common in the interoceptive apparatus, and the neurotransmitters most commonly involved in NSS differ from those typical of canonical synaptic transmission, Table 1. This opens the possibility of intervening selectively in interoceptive pathways, while preserving motor, exteroceptive, or cognitive neural processes. Similarly, the abundance of BBB gaps in the INS may enable the selective access of even relatively large pharmacological molecules, while 1521188, 2021, 6, downloaded from https://onlinelibrary.wiley.com/doi/10.1002/b.202000261